Participants, now we'll start our second lecture, which is on biological control of weeds in India. And our speaker is Dr. Sushilkumar, sir. So I will just briefly introduce our speaker. So sir is having vast experience in the field of biological control. He obtained his MSc entomology from Meerut University in 1981 and PhD from Forest Research Institute, which is in Dehradun. And he started his career as research assistant from FRI Dehradun and later on he joined the ARS service in December 1991. So during his ARS service, he served in Potato Research Institute, Shimla, for about 2 years, and after that he joined the Directorate of Weed Research. So he has worked extensively on biological-based integrated management of terrestrial and aquatic weeds. His most pioneering contribution are for the management of Parthenium and water hyacinth, and he has been recipients of many awards like Ajra Young Scientist Award, DWR Best Scientist Award, 2 times, Fellow of Indian Society of Entomology, then ISWS Recognition Award, ISWS Best Book Award, ICAR Extension Award, Crystal National Agri Award, and many more. So he has served ISWS, Indian Society of Weed Science, as secretary for 2 terms, and also president also, and as chief editor of Indian Journal of Weed Science. So with this brief introduction, I invite our speaker, Dr. Sushilkumar sir, for his talk. Sir, please. Sir, please unmute yourself. Sir, unmute कर लीजिए.
सर जूम में जाके आपको अनम्यूट करना होगा पहले से. यस सर. फिर पीपीटी क्लिक करके जाइए सर. पीपीटी को क्लिक कर लीजिए सर. यस, फुल स्क्रीन कर लीजिए सर. फुल स्क्रीन कर लीजिए. यस सर. ओके.
So thank you very much, Dr. Yogita and the organizers for inviting me for this important talk. So far we were listening from Dr. Nimal, and he has rightly said that we should not say weeds as a threat. But many times we are bound to say that they are threat unless we don't opt for its utilization. So my topic is about the biological control of weeds in India, and we so far by now you are well listened about the problems of different type of the weeds, including invasive weeds. So alien invasive weeds present a major current and future problem in the world, and several weeds have been introduced in India and other countries either by accidentally or deliberately. So alien invasive weeds are recognized as the second largest threat to biological diversity after habitat destructions, and there are different economic impact of invasive pest, and the impact many times it becomes tremendous. And for example, that how an invasive weeds may cause a lot of problem to us may be understood by this picture or this PPT or slides that which we have estimated in India itself. So it is about the Parthenium, a case study. So in general, about 35 million hectares is infested with Parthenium currently. And for management of Parthenium, two hand weeding or two chemical applications are essentially required. And it has been estimated that about 2.5 million US dollar will be required per year by manual labor or 1.6 million by chemicals. Of course, chemical costs become reduced in comparison to manual labor. So these losses may be higher if we take into consideration the aesthetic value impaired by the Parthenium, as well as they cause loss to our biodiversity. So in this slides, you will see that in previous slides, you now understand that even one alien invasive weeds, if it has become real problematic to any country, may cause how much loss. So here there are threat of about 975 species which from different countries. So we should be very cautious about the problematic weeds, and we should try our best that these problematic weeds should not be entered into our country. That's why a very strong quarantine system needs. So now we come after this introductory remarks, then we now come to biological control of weeds. In fact, this biological control of weeds has been accepted as an effective, environmental friendly, sustainable, economically viable, and socially acceptable method of pest management. Of course, there are several methods for the management of weeds, but biological control has been accepted due to their good attributes. And if we talk about what is biological control, then we can say that biological control of weeds involves the use of living organisms to attack a weed population to keep at or below desirable level without significantly affecting useful and wanted plants. So, and times to time, it has been proved that biological control bioagents, they work very nicely in very large areas. Of course, if we talk at a small area, it has been proved that in small areas, their activity remain low because all biological control strategy or its method depends on population buildup of the bioagents. This is the reason that if a large area is infested by a particular weed, and there we have one particular bioagents, then we can get very good results. Now, before going into detail about the what type of bioagents are there, I think it is very important for the participants to know the history of biological control of weeds begin from India. You will be surprised that biological control of weeds in the world begins from India. If you remember that Dr. Nimal has also said about the commercial dye from Dactylopius indicus. So in fact, this commercial dye was used to be prepared by cochineal insects on prickly pear. And that was the beginning. And later on, systematic biological control research started with the establishment of Indian Station of Commonwealth Institute of Biological Control at Bengaluru in 1957. Later on, ACRIP on biological control and weed was started in 1977. And in later years, this Project Directorate of Biological Control, earlier which was known as PDBC, came into existence, which is now called as National Bureau of Agriculture Insect Resources. And in 1989, this Project Directorate of Weed Research came in the form of National Research Center for Weed Science. So now, particularly at this moment, at present time, DWR, ICAR-DWR is responsible for most of the biological control activities against the weeds. How much successful biological control of weeds? Often this question is asked, and proponents of biological control like me, they say that biological control is an effective method. It is very economical and eco-friendly and sustainable. But opponents who does not believe in biological control, they find that biological control, of course, you are talking about biological control, but it is very low, very slow, not able to control all weeds, involve costly and cumbersome process for import of bioagents, etc. And even they say that high rate of failure of bioagents. So, but just to prove that the biological control is not a failure, it is one of the best strategies for the problematic weeds, particularly of alien weeds. Some statistics on biological control needs to be known by the participants. In world, so far about 468 bioagents under 1,555 intentional release has been made against 175 species of target weeds in 48 plant families. So this statistics is very important to understand that how biological control behaves and what are the rate of success of biological control. And out of these all releases, about 63.2% led to establishment of the bioagents. And of the 468 biocontrol agent species, 332, which is about 70.9%, established in at least one instances. So this statistics is very impressive if we talk about the biological control of the weeds. And further, if we wish to elaborate this statistics, then we will find that out of 313 species for which impact could be categorized, about 55% caused medium, variable or heavy levels of the damage. That matlab in the form of impacts of the bioagents. And across all countries and regions, about 65.7% of the weeds targeted for biological control experience some level of control. And if we talk about the countries which are very prominent or which are very active in using bioagents, maybe we can say Australia, North America, South Africa, Hawaii and New Zealand are some of these countries which believe that biological control is very profitable methods for the weed management. And if we see worldwide success rate of biological control in this table, you will find that about out of about all the species, about 426 insect species have been released so far, and medium, variable or heavy impact out of 426 was 218. So this is quite good number, good percentage of the success of bioagents. And out of these bioagents may be insects, mites, nematode, fungal pathogens. And if you will see that even in mite, which we considered is not a very good bioagents, there was 6 mite bioagents were very successful. Of course, in nematodes, we can say that these are not very much successful, but out of 30 fungal pathogens, about 22 fungal pathogens were successful. So this table gives an impression that, of course, opponents, they say that biological control, many times it is a failure, but statistics which have been gathered reveals very positive things about the biological control. And if we talk about the India, maximum degree of success with classical biological control agents was achieved in biological control of aquatic weeds, followed by in crop situations, which is about you can say 47%, and again it is followed by terrestrial weeds. So this statistics also proves that bioagents against weeds are very much successful. And now economic consideration, if we talk, then benefits if we take into consideration, then we will find that about $180 million per annum benefits is caused in USA from the major weed biological program into 20th century. And biological control of Xanthium stumarium, which is also a very big and very new, problematic weeds in Australia. And it was found that in Queensland, when Biogens was released, annual benefits of US dollar of 7.20 lakh was obtained. And in South Africa also a biocontrol program have saved US dollar 276 million in weed control cost. And there are thousands of examples of benefits caused by the Biogens due to their use against the weeds. So now one by one, we will talk about the biological control of weeds in India. And in this series, first of all, we will talk about the terrestrial weeds. And as in beginning, I told you that the biological control of weeds happened to first in India. Of course, that was unknowingly, and that was due to the prickly pear and cochineal insects. You might have heard about the caramine. In fact, it is a sort of red dye which used to be made by the insects interaction with the weed prickly pear. So the first outstanding biological control by insect Dactylopus indicus, which was imported in 1795 from Brazil to produce commercial dye from cactus. So in fact, in 1795, this Dactylopus indicus Biogent was imported from Brazil with the hope that it will produce very good dye. But it acted differently because there was misidentification of the species. So it was instead of Dactylopus indicus, it was species Dactylopus cacti, and although Dactylopus indicus failed to produce superior quality of dye, but it controlled cactus species Opuntia monocantha from India. And another insect, Dactylopus tomentosus, was imported in 1924 from North America, which control another cactus species, Opuntia dilani. So this was the first deliberate example of biological control
सर
of in
स्लाइड
1924.
सर स्लाइड ना स्टॉप हो गया है सर, इसको एक बार फिर से अनइंस्टॉल वो स्टॉप करके फिर से वो करो ना सर।
Moved? Now it is moved, huh?
हाँ, यस 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 सर, ओके सर।
So now actually out of the slides I was speaking and I wanted that participants should know more about the prickly pear and that Dactylopus indicus. So now I come to another weed, that is lantana. I think every participant, they might have heard about the lantana, that how much lantana weed is causing threat to our biodiversity, to our forest, etc. In fact, recently we know that in Chhattisgarh, thousands hectare, lakhs hectares of the lantana forest, which was infested by the lantana, teak forest as well as sal forest, was manually controlled, was manually uprooted to save the forest in Chhattisgarh. So, of course, there are a lot of utilization aspects of lantana, but threat is more by the lantana, and that is the reason that forest managers, they are compelled to manage this lantana by different type of the methods. And one method is biological control. So here, lantana, so far about 9 insect species have been introduced into India, while in the whole world about 55. So in nutshell, if you will found that in the world, initial maximum studies were done on lantana for biological control of this weed. But in India, none of the species cause extensive damage to lantana except this Teleonemia scrupulosa, which this picture you are saying in this slides. This is Teleonemia scrupulosa. In fact, this Teleonemia scrupulosa was also involved in one of the controversies that it may cause damage to one of the very important timber species, that is Tectona grandis. And in 1941, this Teleonemia scrupulosa was imported in Forest Research Institute, Dehradun. But when under quarantine condition, when host specificity was being tested, it was found that it completes its life cycle on Tectona grandis, that is teak. So its culture was destroyed, and that program was abandoned. But anyhow, some weed, some insect, they escaped from the laboratory, and later on in
सर आपका
19...
स्लाइड फिर मूव नहीं हो रहा है सर.
I am not moving, friend. I am not moving. Just I am
Okay, sir.
telling the participant about the lantana camera biological control. I am not moving it. है ना?
Okay, sir.
So,
Okay, sir.
so I was talking about that this biogens, this Teleonoma scupulosa, it escaped from the laboratory, and in 1952 it was recovered. But by that time it was found that it has not touched teeth. So matlab, sometimes we abandon our biological control program with the fear that it will cause damage to other important species. But in nature, seldom it happens. If it is very host specific, so it will not cause damage to other plants. So out of, in nutshells, I can say that out of 9 insect species, so far only this Teleonema scrupulosa is widely successful in India against lantana, and of course it is able to reduce the biomass of lantana during rainy season, but it is not able to control it completely. Now we come to another one of the important problematic weed, Chromolena odorata. In fact, against this Chromolena odorata, which is also one of the very serious weeds of pastures, forests and orchards, particularly in the northeast and south India. And in 1984, Parachutus sedulensolata, it is one of the moth biogens, was imported from Sri Lanka, and it was tested and released in India. Initially, this insects, it caused very good control, but later on it was parasitized by other insect species, and gradually it was found that its intensity of the causing damage to Chromolena odorata are reduced. So now at present its status is partially effective. And in another attempts, we also tried this gallfly Sesidocharas connexa in 2002 from Indonesia. This particular insect species was imported, and it was released in Karnataka, Kerala, Assam, and Chhattisgarh. Initially, it was very effective in particularly Karnataka and some parts of Kerala. And later on, gradually, its effectiveness was reduced. So, at present, we can say that even it is present in Kerala, Bangalore, in Karnataka, and in Chhattisgarh, particularly in Bastar area. But overall impact is, it is partially effective, not very successful biogens. And if we talk about the another biogens against the crofton weed, that is Ageratina adenophora. So we will found that this species is also, particularly this crofton weed, is very problematic weeds in our north and south India, particularly forming dense thickets. And this biogen was imported from New Zealand in 1963, and at present we can say its status is also partially effective. So now we talk about the biological control of Parthenium. Against biological control strategy against the Parthenium, we tried to import 3 or 4 biogens. But before coming to the biogens, I think it is also very important to know about the biology, about the Parthenium, that how much Parthenium infestation level, and in about it was found that Parthenium is dominated in about 22 species. So if you talk the spread of Parthenium, in the left side picture, you will find that it was condition in 1955 to 1977. But by 2025, at present, the Parthenium is present throughout the India. and particularly its presence is very important. If we talk that it has been found in Kargil region of Jammu Kashmir, Andaman and Nicobar, and mini Koi Island of the Lakshadweep. So this is about the some pictures which are showing that how much spread of Parthenium and its infestation level. When because Parthenium is so invasive that it is very hard to managed by different type of the methods, and its seed dispersal into orchards from nearby area, and you see the seeds are very minute and even they are having purpose. This is the reason they are able to spread in wide areas, and they have the ability to germinate throughout the year. These are the attributes which are responsive for its spread, and one of the important thing that Parthenium is responsible to cause health hazards to man and animals. Now we talk about the biological control of Parthenium. So, in fact, attempts have been made to find bioagents, indigenous bioagents, and also attempts have been made to import bioagents from other countries for the biological control of Parthenium. Under indigenous insects, this Nupsera species was found, and infestation level was found 10 to 95% during rainy seasons. So, in some pockets of India, the heavy infestation of this Nopsera is found. But Nopsera species is not host specific. This Nopsera, particularly insect species, infests other asteraceous plant species. So we found that, of course, this Nopsera is very good bioagents against Parthenium, but because it is not host specific to other plant species, so we are not promoting this bioagents against Parthenium. And if we talk about the exotic insects, tried was made to import flower feeding weevil Smicronix lutulentis, which could not be multiplied in the laboratory. And now again, attempts are being made to import it, and testing, host specificity testing is going on in India at NBAIR Bangalore. And stem boring moth Epiblema stridulata was also imported, but it was found that it feeds on Niger, so it was also abandoned. And host, another bioagent was Zygogramma bicolorata, which you might have heard by the name of Mexican beetle. So it was found very useful and host specific and safe bioagent in spite of its initial controversy on the feeding on sunflower. After successful laboratory test, it was released in the environment. But later on, some scientists they found that this bioagent feeds on the sunflower. So once upon a time in, you can say by 1990s, there was big controversy that the concerned scientists who have imported this bioagent, they have made a very big mistake by introducing it into India, because it is feeding on sunflower, which is very important oil yielding plant in India. But later on, due to that controversy, one big fact-finding committee was made against this bioagent, and a lot of research, coordinated research was made throughout the India, and it was proved that it only nibbles the sunflower plant, and it may never be a pest on sunflower. So after that fact-finding report, its intentional release was made possible by the Indian Council of Agriculture Research. Now we can multiply it, we can release it anywhere in the India. So you in this picture you see that how much large area it is able to control. In this picture you see, and in the lower picture you see, and host specificity also you see, because in one of the sorghum crop, you see amidst the sorghum crop there was the infestation of Parthenium, but it has almost defoliated and killed the Parthenium, but it has not damaged the sorghum. So it is very host specific, and this is the reason that we are promoting this bioagents again, one of the very dangerous weed, that is Parthenium in India. you see that how it is able to defoliate very big Parthenium. These are the bioagents, adult bioagents of Zygogramma bicolorata. These are the grubs of Zygogramma. And you see that gradually they will nibble it, they will defoliate, they will completely defoliate the Parthenium plants. Even very big Parthenium plants, they will completely defoliate. And if they get opportunity to find a small Parthenium, they will completely kill it. In this gradual sequence of the picture, you will see a small Parthenium plant was invaded by larvae and adults of the Zygogramma, and gradually it was completely killed. And if we see the overall impact of Parthenium in about fourth year or third year, you will see the large area which was early infested with Parthenium only, gradually with the act of biological control, with the act of this bioagents, other vegetation started to come. And by the third year, Parthenium become very less. And by the fourth years, almost that whole area was was occupied by another plant species. That was, of course, in this case, it was Cassia tora. So you will see that how gradually this bioagent is able to control completely Parthenium. That was the reason that now Parthenium, we are using these bioagents throughout the India against the Parthenium. If we talk about the economic benefit by the maxibitan beetle in an study which was done at DWR, we found that by the fifth year, release of Mexican beetle in Jabalpur, it caused benefit of US dollar of 10 million. It means a huge benefit. And now at present, if we talk about the economic consideration of this Mexican beetle, in one of the estimates, we calculated that every year about 6 billion rupees benefit is being caused by this bioagent alone throughout the India without man efforts. And whenever we will try to release this intentionally, we will find that its potential or its damage activity will be increased manifold. And this was the reason that ICAR-DWR is making attempts to popularize these bioagents by distribution throughout the India. And initially we used to send to all agri centers and Krishi Vigyan Kendras, and that was the reason that now at present in whole throughout the India this bioagent is has been established. And recently our Directorate of Weed Research, they have observed this 20th Parthenium Awareness Week. For last 20 years, regular activities being done by DWR throughout the India involving ACRIP and Krishi Vigyan Kendra, etc., to make people aware about the Parthenium and the ways of its management. So now, keeping this in view, that biological control-based integrated management of Parthenium was attempted in Northeast region also under one of the DBT projects, and we found that if we talk about the Parthenium Northeast, we found that severe Parthenium infestation was in Assam and Nagaland. Of course, in Sikkim and Meghalaya, it was less. And we also released bioagents there, and we found faint recovery of these bioagents in Assam, particularly in Guwahati region. And now, after this exposure of terrestrial biological control of weeds, we come to the biological control of aquatic weeds in India. Of course, you all know that aquatic weeds are also one of the biggest problems in water bodies. And if we talk about the major aquatic weeds of India, we will, out of 140 aquatic weeds, about 12 weeds are very important. And if we talk and if we see the pictures, some important aquatic weeds are these: Ceratophyllum, Pistia, Chara, Hydrilla, Nazas, Natella, etc. And you, all the participants might be, I think, recognize this weed. This is water hyacinth. This is called one of the worst weeds of aquatic bodies. And bioagents and biological control attempts have been made against this water hyacinths throughout India. And we all know the problems caused by aquatic weeds because they, particularly if we found that they are able to reduce the water heavily, about because 2.2 to 4 times more evapotranspiration caused with the water hyacinth. And this is the reason that wherever water hyacinth will be there, there will be dryness of those problems due to the heavy loss of water through evapotranspiration. And you see, these are the some of the pictures which are showing that how much problematic this pistia in Bhima River near Pune in Maharashtra, and in Uti Lake. Once upon a time, this water hyacinth was very much problematic. But now this Uti, because it is a tourist place, now they have taken this as one of the major activity to remove water hyacinth from the Uti lakes. So this is the reason that now if you will visit, you will not find, or you will very less find this water hyacinth in Uti lake. And in rivers, many lakes are found infested throughout India through this water hyacinth and other weeds. You might have heard about the Dal Lake. Dal Lake is also infested with aquatic weeds. Means submerged and floating, both aquatic weeds are there. And this one picture is showing that how much irrigation canal has been infested by water hyacinth. Ponds, rivers, canals, dams, they are infested by the water hyacinth. This picture is showing that how this blanket weed, submerged weed, is has infested one of the canals in Madhya Pradesh. In UP, this water hyacinth has infested many water bodies. Now, what are the options? Of course, there are different type of the options for the management of this aquatic weeds: preventive, manual, chemical, and utilization. So, of course, because this topic is about the biological control, so we will emphasis on biological control activities only against these weeds. So we talk about the water hyacinth biological control. You see, this is one of the bioagents which is called Neocatina species, that it is able to control huge infestation, severe infestation of water hyacinth in very large bodies. And these are the pictures that how gradually it is able to control water hyacinth. And this is another picture which is showing biological control of water hyacinth. And this is another picture of biological control of water hyacinth by Neocatina species. Now, one of the recent in successes, if we talk, I want to share with you my experience about the Salvinia monesta by one of the bioagents, Ritobacus salvini. In fact, this Salvinia molesta weed, it was not earlier considered a important weed in central India. But the recent survey revealed that it has invaded central India as well as North India. So we started to work on this biological control against this Salvinia molesta, and we released bioagents in different aquatic bodies infested by Salvinia molesta. And gradually, we also did a survey of Salvinia molesta, and we found that this Salvinia molesta, how it has infested lakes and water bodies in Madhya Pradesh, in Haryana, in Maharashtra and Chhattisgarh. And this is one of the lake, very big lake in Talpuri in Durg, which was heavily infested by water hyacinth. And here this bioagent was released, and gradually it was controlled. So occurrence of Salvinia molesta, if we see this picture, we will find that earlier it was dominant in South India, but now gradually it has invaded central India as well as North India. So its threat is being observed by weed scientists. And this is the reason that we have taken this biological control program in bigger way at our Directorate of Weed Research, and we have released this bioagents in different water bodies. of course, if you, after release of these bioagents, what happens about its population? You will see in this picture that how gradually population increase from zero, then it will go to its pinnacles, highest, and then it remains almost statistics, and gradually then population begins to decline. But this happens with the control of salvinia and water visibility. Here you see earlier the whole pond was infested with water hyacinth, and water visibility was zero. But here water visibility is around to about 95 to 98%, and that happens within about 15 to 19 months. So this is how this bioagent works. And the biological control of weed is related to population buildup and then control of the concerned weed. And in this picture, you will gradually see that how in one of the ponds in Katni of Madhya Pradesh, that after release of the bioagents, gradually, if you will see all the pictures, you will find that in July 2020, this was the picture, and in June 2021, complete whole this water body is clear. And recently, our team went to there and found that no further Salvinia molesta has invaded in this pond. It means that it is cost effective as well as sustainable, because only one of the releases have made complete control of this Salvinia molesta. And these are the other aquatic bodies infested with Salvinia molesta. And these are our efforts of releasing these bioagents in different water bodies of Chhattisgarh, Maharashtra, and Madhya Pradesh. And our efforts were considered by the media also, and Times of India magazine and other magazines, they covered this event. And these are the success story was also put up on the ICR websites, and Gaon Connection, one of the popular magazines, they also covered our story. So you see that how Panchmani Lake was completely controlled and other bodies were also controlled. Gadchiroli is known for the Naxalite activity, but it was also once upon a time known for the activity of the Salvinia molesta. Very 3 or 4 big lakes in Gadchiroli, they were infested by Salvinia molesta, where this bioagent was released, and now no water body is infested with Salvinia molesta. And like this, in Sarni, in the Baitul district of Madhya Pradesh, we control about 3,000 hectare water bodies, which was completely infested by the Salvinia molesta, and it has been controlled completely by the Cetobacter salvini, that bioagent. And this particular success of biological control of Salvinia molesta was covered by the one of the popular magazines. You might have heard this Down to Earth. The representative of the Down to Earth, they went there in Sarni. They interviewed the people. They interviewed the Thermal Power Corporation authorities, and then when they found that yes, really this did a wonderful job, then they covered this story in their magazine. And you see that estimation required by population for activity of biological different type of the research activities was done at DWR. And now we can say that about if we release about 22,800 bioagents, adult bioagents in per hectare area, then it will take about 15 months to completely control the Salvinia molesta in that particular water bodies. Even if we if we release lakhs of beetles, there will be not too much benefit of the time. So, but release of 22,000 will be economical. So, like this, biological control is very successful in Kerala also. And apart of this biological control by the insect bioagents, some grass carps, they also do wonder controlling particularly submerged weeds. Of course, the attempts were also made to integrate chemical and biological control, and we found that time may be reduced taken alone by the bioagents if we integrate chemical control. but always there is question that whether we should chemical in the water bodies. So of course, but in about 15% area and very less area, if we cover through the chemicals, then certainly we can reduce the time taken by the bioagents alone. Of course, some studies have been done by the microherbicides, which was developed by the AG Biotech Hyderabad against the Parthenium, Lantana, and water hyacinth. Of course, this study is still underway, and but we found that it was that bioherbicide was effective against Parthenium and water hyacinth, and of course, it was not very much effective against Lantana. Work is going on by the AG Biotech against this. And we also tried to find the climate change effects of bioagents, particularly Zygogramma bicolorata on Parthenium control, and through various studies on dry weight and population dynamics of the bioagents, we found that there may be effect of the climate change on the bioagents to control the weed, but that effect is not very much. In nutshell, our result indicate that the reproduction efficiency of Zygogramma bicolorata is likely to be reduced as the climate change, despite increased feeding rates exhibited by grubs and adult beetles. So it is a win and win situation. Of course, with the climate change, increase in CO2 level, Parthenium, particularly this bioagent insect activity will also increase, and they will equally effective as they are effective at present. So if in nutshell at the last, if we say that what is the current status of biological control in India, that was also covered by Down to Earth in the form of an article promising agents in the Down to Earth magazine of September 2024. So in nutshell, we can say that so far about 32 bioagents have been introduced in India against the weeds. 4 failed in laboratory tests and abandoned, and 5 could not be recovered. So 5 or 4 plus 9 in such a way are not very much effective. Out of 32, about 23 bioagents were found established, out of which 6 provided excellent control, including this Neocatena species, Cytobegus species against Salvinia, and Zygogramma bicolorata against Parthenium, and of course this against Cromolena odorata and this Crofton weed. And if we take overall situations of the biological control efforts in India, again, if you remember that aquatic weeds success was in 55.5%, while in terrestrial weed it was around 28%. Of course, if we talk about the crop pest, it was around 46%. So challenges are, of course, there, search of host specific agents, control of woody weed species, long-term efficacy of bioagents. These are the challenges, because earlier you might have heard that some of the species, their efficacy becoming reduced. So we long-term efficacy or maintaining of the long-term efficacy of the bioagent is also a challenge. And if we talk about the perspective, then of course to opt biological based integrated management, find out suitable release techniques in heavily infested water bodies, introducing more successful bioagents against problematic weeds in other countries, and about that efforts are being made by the DWR ICR. Thank you very much. So with this, I think my topic is over on biological control of the weeds. Yeah. So
Okay,
now,
thank you, sir. Sir, now we'll take questions from participants.
sure.
So participants, you can raise your hand. Okay, Dr. Pratibha Sri Sunkara, you can ask your question, unmute yourself. We are not hearing. Dr. Pratibha? Okay, Mr. Shailendra Singh Kushwaha.
Sir, I want to discuss you with regarding the lake in Sipuri, which I have visited recently in month of June. where this Iconia Crosby has invested very badly the Supri Lake in Madhav National Park. And you have visited that lake also in 2013-14, I think. But there still it is infested that lake, and the lake is very much infested with that weed. And what is the solution for that? Because it is not available, your bioagent is not available there. I have suggested the DFO taking care of that lake to utilize this Neochetna beetle, but they have said, they told me that it is not available in the that much of quantity which we required here. So what is the solution for such type of situation?
Yeah, Dr. Shailendra, in fact, I visited this Sipuri lake in 2023 after my retirement from ICAR-DWR. So what I found that there is great possibility, there is greater possibility of biological control there in this lake in Madhapuri, because this lake, it is a sort of twin lake. So this lake is also linked with one of the another lake which lies in the city, and through the water channels it is connected with this
Exactly.
forest national
Yes,
park.
yes.
And I have suggested them to utilize, because what I saw that this lake is very suitable for biological control activity, because this some specific conditions are required for biological control. Means matlab one activity is there, that concerned pond should be perennial pond, or water body should be perennial. Means matlab water should remain throughout the year. Why this is so? Because if that water body will be dried, then there will be no population buildup of the bioagents. And I found that this lake in
National Park, National Park.
National Park. So it is very suitable, and I suggested them that there is possibility of biological control of these feeds, and they can talk with the authority of ICAR-DWR. And in fact, I think they did not approach to DWR. Rather, later on, I learned that they approached one of the State Forest Research Institute, that is SFRI, in
Okay.
Jabalpur. So
Okay.
they wanted SFRI that they should, because I think Madhav National Park comes under the Madhya Pradesh Authority, and they might be, I think, very congenial with the state research department. So they approached me, and I suggested them to make a project, research project, therein they can release the bioagents. But I think later on I did not hear from SFRI also, and I think there is a lack of interest by the authorities there. In nutshell, I can say only this one. Otherwise, that water body is very suitable for biological control.
Yeah, sir. And this case is also taken care by the MP High Court branch Gwalior. Bench Gwalior, after one person has filed a public litigation PLI regarding this infestation.
Yeah.
So DWR must take some serious step to take the case study for this to handle this situation, because this lake was very beautiful earlier when I visited here, the Sipuri. Now it is in very bad condition, sir. So
Yeah, I think
the
if
things,
you are, yeah, please, Salemji.
sir.
If you are having contact with them, with the DFOs and forest authorities, then you please kindly convey DWR interest, that they are always happy to collaborate with them, provided matlab, they give some funding. Otherwise, without funding, it will not be possible by DWR also to work there constantly.
Okay, sir. Thank you, sir, for nice input, sir.
Yeah, thank you.
Dr. Pratibha, you can ask your question. Ms. Megha Tiwari.
Good afternoon, sir.
Yeah, afternoon.
Sir, my question is that the farmers who are suffering from this weed problem in their pond areas, how they can easily get this biological pest from where they can purchase it. Means the local farmers, they are not able to purchase it in local market. So I need a particular area from where I can tell the farmers to purchase. If they are having any problem, then they have to purchase zygogramma beetle, then where to go, where to approach. So they must have a perfect address and perfect contact number, sir.
Yeah, Dr. Tiwari, your question is very pertinent. In fact, always such persons, particularly farmers and interested persons, they want to know from where we can get these beetles.
Yes, sir.
In fact, by now, this beetle, particularly the Zygogramma bicolorata, is available throughout the India, provided if farmers they are able to search it, and particularly during rainy season, if if they will visit such area which is heavily infested with Parthenium, and if they will overturn the Parthenium leaves, then they may find these bioagents there itself, and they can themselves collect, and then they can release from such area where these bioagents are in abundant, and then they can release in those area where these bioagents are in less number or not present. So this is a sort of augmentative behavior or augmentative biological control. And of course, even if they don't want to make such efforts, they can approach our ICR Directorate of Weed Research. They can,
Okay.
I think they can purchase from here itself.
So what will be the cost for the farmers?
I think around 2 rupees per adult beetle.
2 rupees. Okay.
Per
Thank you, sir.
adult.
Okay,
Yeah.
sir. Okay, sir. Thank you, sir.
Miss
Yeah.
Kavita Sagar, you can ask.
Yes, ma'am. Good evening, sir.
Good evening.
Yeah. So through your presentation, I learned that there are certain challenges to identify suitable or appropriate bioagents. Right,
Yeah.
sir. So I'm interested. Yeah, I'm interested to know how to overcome these challenges.
Yeah. So if you have, if you recall, I was telling about the indigenous bioagents against Parthenium. That was the
Yes.
Nuxera species. In fact, each and every weed is infested or is attacked by some sort of insects. This is very common. It is very natural. As we found some insect species, they attack our crops. Likewise, every weed species is being attacked by some sort of insects. But if we talk that what type of insect species will be suitable to act as a bioagent, to act as a effective bioagent, this is our in fact challenge.
Yes, sir.
For example, against Parthenium, we found that this Nuxera species heavily infest Parthenium during rainy season. Almost out of 10 plants, 5 plants we used to find infested with Nuxera species. But because Nuxera species is not a very host specific, so we did not dare to use this indigenous species for the biological control agent. So always this is a challenge that we should find a very host specific bioagents. So this is the reason that against most of the alien invasive weed species, we are bound to import effective bioagents from those countries in which that particular species has been originated, or from other countries where these effective bioagents have been imported and they have been found effective against those weed species. So more and more means import we will do, more and more success we will get. This is the, I think, thumb rule of the biological control, particularly at present in India. If you recall that only 32 bioagents have been imported so far in India. While if we talk in Australia or in other countries which are actively involved in biological control, have even 50, 50, 60, 70 type of the bioagents they have imported from other countries, or the countries from the origin of that weed species against that problematic weeds in their countries. So bioagents, particularly this alien bioagents, will be more effective under the present circumstances. There will be no use of means matlab release of indigenous bioagents, and this has been proved again and again through systematic research efforts also.
Okay. Because as I learned, because most of the weeds present in India, especially Parthenia, Mycorrhiza, Lantana, all these are alien invasive weeds. So in order to control by using the biocontrol agents, we need to get the alien biocontrol agents.
Yeah.
Is that what you say, sir? Okay.
Yeah.
Yes. Thank you. Thank you, sir.
Yeah. Thank you.
So we have one question in our chat box, question and answer box. You can see, though answer is given, but you can elaborate that one, that release of biological agent to control weed is effective and efficient. If we take a hypothetical situation, once the host weed diminished, what will be the fate of those pests? Is there any chance they search for another host weed or will affect the crop?
Very interesting questions, and always this comes into the minds of the users of bioagents. And as he has asked that what will happen once, for example, if there is a win-win situations. Hypothetically, if we imagine that once a weed species is completely controlled by the bioagents, what will happen to the bioagents? So it is very interesting that if weed will be vanished or weed will be finished, bioagent itself will be finished. If we recall our one of the slides of the population dynamics of the bioagents, where we found that initially its population become very low. Gradually it increased and reached at highest level, and sometimes it remains statistics there, very stable there. Particularly, it depends on the weed species or weed species characteristics, that how much area, how much time it should remain there static. And gradually, what will happen that it will population of this weed will diminished or vanished, and gradually population of the bioagent will also decrease, and ultimately with the complete control of the this weed, bioagent itself will also be vanished. So there is always this situation occurs, but in nature it is the hypothesis, but in nature it never happen. Sometimes it nook and corners this weed also remain, and bioagents on that weed also remain. And when this weed try to overcome or it increase, then gradually this bioagent also increase. So this is the reason that bioagent is sustainable, because when weed will try to increase its population, bioagent gradually population of bioagent will also increase. So this happens in nature. I think it is clear to you now.
So, okay, sir, thank you very much, sir. We know the biological control of these weeds found its way in India because of you, because of your efforts only. We have many success stories of biological control of weeds, especially biological control of aquatic weeds, Salvinia molesta, by this bioagent Siltobacus salvini. So thank you very much for giving very insightful talk. Thanks for accepting our invitation on behalf of organizing committee and on behalf of participants. I thank you for your insightful talk. Thank you
very much, Dr. Yogita. I also thanks to organizers for this opportunity given to me. Thank you very much again.
Thank you, sir.